Namaste, this is your Charanya. Today we are going to discuss about plants from class 6 to class 10. In class 6, we have parts of a plant. This is a plant. This is our ground level. The part of the plant above the ground level is known as shoot system. In the part of the plant below the ground level is known as root system. First, let us see into the root system. There are two different types of root, taproot and fibrous root. The two different types of roots are taproot and fibrous root. They have some differences. This is the main root. From this main root, other roots branch out. The roots which branch out are known as lateral roots. So a taproot consists of a main root and lateral roots. Whereas in a fibrous root system, all the roots are of the similar size. All the roots are of the same size and they also look bushy. Now let us see about the cotyledons. Now what is this cotyledon? Have you seen a seed picture? First it will be having a seed coat. After we remove the seed coat, we will find two cotyledons. The two cotyledons are the baby leaves which prepare food for the embryo. Now, if a plant has one cotyledon, it is known as a monocot. Mono means one. Cot means cotyledon. Monocot. If a plant has two cotyledons, it is known as dicot. Di means two. Cot means cotyledon. So it is known as a dicot. Fibrous root has monocot. If you see in fibrous root, if you take one strand of a root, all the roots will be of the similar type. It means only one cotyledon, monocot. Here, it will be the reverse, dicot. So in fibrous root, all the roots are of the same size. It is a monocot. In tap roots, it has the main root, and lateral roots, it is a dicot. Now let us see into the modification of roots. Before going into the modification, let us complete the functions. It absorbs water and minerals from the soil. It absorbs water and minerals. These are all the different types of minerals present in the soil. It absorbs water and minerals. And also, it anchors the plant into the soil. Anchors in the sense fixing the plant into the soil. It also, the roots, to fix it, it has to go deep. While it's going deep, it clings. It holds on to the soil and prevents from soil erosion. In plants like carrot, radish, turnip, these roots store food. At last, we will be seeing the modification of roots. First, let us complete the parts of the shoot system. This is all about roots. Next, the part of the plant above the ground level is known as shoot system. In shoot system, we will be having a stem, a flower, fruit, leaves, buds. This is also one bud. Buds, etc. First, we'll go see into the stem. Stem is the main axis. What is this axis? Axis means the central structure. For this structure, all other parts come out. So a stem is the main axis of the shoot system. And also it grows towards light source. Above the ground, obviously light will be there. So it grows towards light source. Whereas roots, in soil there's no light. So it is going away from the light source. It goes towards the light source. Now we know what is a stem. Let us know the different structures present on the stem. The point at which a leaf touches to the stem is known as node. And the space between one node and another node is known as internode. It is known as internode. And suppose there is a stem, here's a leaf. At this angle is known as axil. If here any bud is present, it is known as axillary bud. And the bud, bud, bud present at the top of the stem is known as apical bud or terminating bud. We know what is an internode, we know what is a node, axil, axillary bud, and apical or terminating bud. After this, let us go into the leaves. This is a diagram of a leaf. Let us know all the structures of the leaf. This will be our leaf base. 1 minute. Leaf base. And what are these? These are the veins. You can see these are the veins. And the middle vein is known as midrib. It is known as midrib. And this is a vein, and other veins which are branching out, smaller ones, they are known as veinlets. See, here also in root they branch out. They are known as rootlets. Those rootlets are known as lateral root. Here also when the vein branches out, they are known as veinlets. It is no other name is there for this veinlets. It's just veinlets. And after we see into the midrib veinlet, this is known as a stalk. Staklk. This stalk is known as a petiole in a leaf. It is a petiole. What is a petiole? This one is a petiole. Now, whichever leaves have this petiole, they are known as stalked leaves. What are they known as? Stalked leaves. If they don't have this petiole, they are known as sessi, sessile leaves. Sessile leaves. Stalked leaves and sessile leaves. And venation. Venation means the arrangement of veins is known as venation. The arrangement of veins. Here, all the veins are like a net-like structure. So, it is reticulate venation. Reticulate venation. And another type is that all veins are parallel to each other. For suppose, this is a grass plant leaf. Here, all the veins will be similar to each other. They will be parallel to each other. So, it is known as parallel venation. We have two types of venation, reticulate venation and parallel venation. Now, reticulate, net-like. So, here also you can see from the main root, other lateral roots are branching out. From the midrib, all veins are branching out. So, reticulate venation comes under taproot system. Reticulate venation comes under taproot system. And all are parallel to each other. That's clear. Here, see, everything are parallel to each other. All are similar to each other. So, parallel venation means fibrous root. Parallel venation means fibrous root. Now, in this fiber is there. Fiber means how? It will be like a thin strands. Here also thin strands. So, fibrous root and fiber, you can remember like this. Now that we have completed venation, what does these leaves perform? Its functions. First thing is photosynthesis. That's the common function. Photosynthesis, it is the process by which these leaves prepare their own food. And they're also known as food factories. Factory is a place where something is made. Food factory, as they're making food. And after food factories, photosynthesis, transpiration. At the lower sides of the leaf, suppose this is a leaf, at the lower side there are many stomata. This is a diagram of a stomata. Wait, I'll name it. Stomata. So the stomata is like a tiny pores through which gases can go. See, this is a stomatal opening through which gases can go. These two bean-shaped cells, they are known as guard cells. What are they known? They are known as guard cells. These are the epidermal cells. Now we know stomatal opening. So this stomata facilitates what does it facilitate? Exchange of gases. So it facilitates exchange of gases during photosynthesis and respiration. And also it performs a major thing, transpiration. Wait, let me list out the points of transpiration. We'll erase this difference. Now in transpiration, what does this transpiration mainly mean? So transpiration is the process by which leaves lose water. So water gets evaporated from the stomata. It releases water outside. Why do leaves transpire? It's a very essential process. Why do they transpire? To give a cooling effect. First thing, we also sweat. It gives a cooling effect to our body. So they'll also lose water. It gives a cooling effect for their body. And also one more thing is for transpiration is, if you see the water and minerals absorbed, it will go upward. You saw? It will go up. Why? What force is pulling them up? Tell me what force is pulling them up? Transpiration pull. By using transpiration pull, all these water and minerals from down, they'll go up. Now we have seen about transpiration pull also. So it helps in transpiration pull, loss of water, so that it gives a cooling effect, and it helps in maintain the balance of water. So these are the major uses of water. Now let us one more time recap the functions of a stem. Stem, it is mainly like a transport system. So stem transports water and minerals from the root, and also it transports the food prepared. Some stems even store food like potato. And what else can we say about stem? Storing food, transport water and minerals. It also supports the other parts. So these are the functions of a stem. We have looked into the functions of a leaf, function of a stem, function of a root. We completed venation and types of roots. Now let us see the modification of roots and classification of plants. We are going to see the modification of roots and classification of plants. First we'll go with the modification of roots. Modification. The main thing here we can see supporting roots. Supporting roots and clinging roots, breathing roots, supporting, clinging, breathing. That's it. So in supporting roots, from the branches, these roots arise. In a banyan tree, if you see, in a banyan tree, these roots give support for this tree. These roots are growing from the branches, they're supporting it. They're known as supporting roots. And in sugarcane, at the nodes, we can see roots coming out. So they are known as stilt roots. Stilt roots. So this stilt roots provide support for this plant. And clinging roots. What are these clinging roots? Mainly for climbers. If you see, this is a climber. 1 minute, I'll draw the picture. This is a climber. The roots will penetrate into the wall cracks, or it will penetrate into the supporting. Suppose the stick is supporting, so it needs that clinging roots to cling upon it. These are clinging roots. It look like this. Clinging roots, supporting roots. In mangroves, these mangrove plants grow in wetlands. So in this wetlands, the roots needs oxygen. It can't take dissolved oxygen. So what does it do? It grows upwards. So that in search of oxygen, atmosphere and oxygen it can take. So it will be growing upwards for breathing. So they are known as breathing roots, clinging roots, supporting roots. We have completed modification of roots. Next, let us see classification of plants. There are major 5 classification. Which are those? Classification of plants. Herbs, shrubs, and then we have trees, we have climbers, creepers, now in herbs, they have green tender stem. Green tender stem. We'll check into the stem pattern. And then sometimes they will have branches and sometimes they won't be having. An example is that tulsi plant is a herb. Grass also is a herb only. Now in shrubs, they will have woody stem. Woody stem. And the branches will arise from the base of the stem. From base only branches will arise. For shrubs. Trees, the stem is very thick. In a stem, very thick stem, it is known as trunk. So in this trunk, The branches will not arise from the base. After certain gap, it will start growing, not from the base of the stem. And in climbers and creepers, they have one feature in common. They both have weak stem. And climbers, they can climb up as they don't have heavy fruits. Whereas in creepers, they can't climb up. They're having heavy fruits, such as watermelon plant. Can it creep? No. Sorry, can it climb? No. But it can creep upon the ground. And in climbers, example is grape, money plant. It will take the help of other sticks or any walls and start climbing. Climbers, creepers, trees, shrubs and herbs. We have learnt about the classification. We completed modification of roots. Types of roots we learnt, which is taproot and fibrous root. We saw the differences. Function is that it anchors the plant into the soil. It absorbs water and minerals. It stores food. In stem, what are the functions we saw? It transports water and minerals, it transports food, it supports the plant and stores food. In leaves, it facilitates photosynthesis, it helps in transpiration, it is a food factory, and sometimes it stores food like spinach, mint, etc. About flower, in 7th class reproduction we will see. Now we have seen about leaves also. And the one more important thing is perennials, biennials. This is just like a lifespan of plants. If a plant lives for one season, it is known as annuals. Annuals. If a plant lives for 2 seasons, it is known as biennials. It is known as biennials. And if a plant lives for many seasons, it is known as perennials. Annuals, biennials and perennials. So these are the 3 different types of plants on the basis of their seasons. For how many seasons will they be living? And now we are going to see some facts. Banana is the largest herb. And one more fact is that weeds. Weeds are unwanted plants that grow, and also it competes for shelter, nutrients. So generally we'll be plucking out the weeds from our fields. We have covered all. We have even learned about, wait, we forgot a very important concept, which is germination. Suppose this is a seed. From a seed to a seedling. And some can even grow into plants, baby plants. So the process of a seed growing into a seedling is known as germination. Now let us see some structures. In a seed, the outer covering is known as seed coat. And after we see the seed coat, we will be able to see the cotyledons. Cotyledons are the baby leaves which prepare food for the plant or food for the seed. And then will come a embryo. Embryo is the baby plant. Embryo. So these are the different things in germination. We completed transpiration and germination. In class 7, we are seeing reproduction. What do you mean by reproduction? Re means again. Production means produce. To produce again. Reproduction is a very important cycle of any organism to continue their birth, to continue their generation. When plants needs to grow, they have to keep their generation or their organism on earth. So they have to reproduce. In reproduction, there are of 2 types. Asexual reproduction, sexual reproduction. In asexual reproduction, again there are 2 types, natural, artificial. First, let us start with asexual reproduction. It is also known as vegetative propagation. In this vegetative propagation, all the vegetative parts of a plant, such as a stem, leaf, roots, everything are used except flowers. Now, in vegetative propagation, it's natural and artificial. In natural, we have bulbs. Stem, that bulged stem of onion is known as bulb. That if you plant an onion bulb into the soil, it will again grow into a new one without using seeds. So this is also asexual reproduction. So that is using bulbs. Tuber. Tuber means potato. Potato is known as a tuber. That using buds, that eyes they'll be having. Potato has eyes. So if you plant a potato, from that eyes of a potato, new plant will grow. New offshoots, new shoots will grow. So this is also one method. It is also known as tuber. So At the natural, we have bulbs, bulged stem, and tuber, it is also a type of stem, and runner. Strawberry has stem runner. What is that does is that stem, it will be growing. Suppose this is a stem. Another branch will grow like this. Here again one more will start growing. This is a stem runner. It looks like running. So it's known as stem runner. And then from leaf also. Bryophyllum. This is a very special type of leaf. From that leaf, some more leaves will generate. A new plant can be grown only if we plant the leaf. Don't need the flower. With planting the parts of a plant only, we can reproduce using asexual reproduction. Natural we have covered. Roots is left. Roots, if you plant a carrot root, it will still grow without using any seeds. So carrot root, a tuber, that is potato, bulb, onion, runner, strawberry, leaf, bryophyllum. This is natural vegetative propagation. In artificial spelling is wrong. Artificial vegetative propagation, we have layering and grafting. Now, what is this basically layering? Suppose this is one stem. This is a stem. Now that branch, we will bend it into the soil. Again here we'll tie it into the soil. Again a new plant will grow. Then we'll cut off the relation. So another we are growing using layering. This comes under artificial vegetative propagation. Layering is done like this. And now grafting. There are two methods in grafting. I'll tell you both. Suppose there's one plant growing. You cut off a bit bark and another plant is growing. From this also you cut off bark. You attach them with a rope. The one is called, this is known as stalk and this is known as scion. After some days when this branch starts growing, you can cut off the relationship with this plant. And again if you cut one branch, still the plant will grow healthy. So this is a part of artificial vegetative propagation. This is one type of grafting. Another type of grafting is that in a plant, suppose this is the stem, we cut it deeply. Gap is left. It is stalk. Scion is that another part of a stem. You insert here. You again tie it with rope. This is the scion. This is one more type of grafting. Let us revise the before one also. Here's a plant. 1 minute. Here is a plant. It's growing. We cut off the bark. Here is one more plant. We cut off the bark. We attach it. And then you can cut this side and this side. The plant will be growing. This one I like draw it a bit more big. This is a plant. It is growing. One more plant is there. Suppose this has disease resistance. Majorly they'll be using these type of methods and all in some plants which has one quality and other quality. With both these qualities you want a good quality plant. So it has disease resistance and this has good yield. They'll join these two plants. That time they can get even better plant. So here we are going to cut some bark. Peel off some bark. Here also we are going to peel off some bark. Attach it. A new leaf will start growing. After that you can cut off the relation with this plant. And also if you cut, again it will grow into a proper plant. By using this artificial reproduction, one tree is there. That tree has 40 different types of fruits. Wonderful, isn't it? Now we have completed artificial. In artificial, when we are using these methods, when we use a bud, and one more thing is that cutting. Suppose this is one plant. In this plant, we cut a part and we attach it with a bud. That is known as budding. And we completed grafting, budding, layering as sexual reproduction also. Let us go into sexual reproduction. In sexual reproduction, a flower is the main place where this type of reproduction occurs. it requires two parents. What are these parents? A male one and a female one. Here, this is a leaf. This is a flower. We label the parts. The bulged part is known as thalamus. Thalamus. And this one, it is known as pedicel. And these leaves are known as sepals. Another name of sepals is calyx. These leaves are known as sepals. And we have one more thing, petals. This is the petal. This red attractive part is the petal. Now, 1 minute. This filament type. This one is filament. This is a filament. This is a long thread-like structure. It is a filament. And this sac-like structure, it is an anther. This anther and filament both come under male reproductive part. Anther and filament both come under male reproductive part. Now this male reproductive part has two names. Stamen. Stamen. Actually the name itself says male reproductive part. And androecium. Androecium. Androecium and gynoecium for a female. Androecium and stamen, they are the male reproductive part. Let us go into the female reproductive part. This one, it is known as stigma. Stigma. And then this long tube, it is known as style. Style. And this part, this is ovary. Ovary. The seeds you can see now, they're ovules. Ovules, ovary, style, stigma. Style, stigma, ovary. These three things. Style, stigma, ovary. These are the three things of female reproductive part. And in this ovary, we have ovules. Style, stigma, ovary. Now, female reproductive part has two names. Pistil. Pistil. I'm sorry. Pistil. And also one more name is gynoecium. Let us recap the parts of a flower. Thalamus, the bulged part, pedicel, the leaves, these leaves, they're sepals. Now, the leaves just below the flower, they're sepals. The attractive part, they're petals. Now this one, the central structure. Stigma, style is the tube, ovary is this part. In this ovary, we have ovules. The central structure, it is the female reproductive part, also known as pistil and gynoecium. We completed this part. Let us go to the male reproductive. Male reproductive consists of anther. This is the anther, which contains pollen sacs. After that, we have filament. Filament and anther, both are stamen, male reproductive part. Now that we know all the parts, let us see pollination. This is the first step of reproduction in a sex bud. First thing is, this anther have pollen grains. What do these anther have? Pollen grains. Now these pollen grains has to reach stigma. These pollen grains has to reach stigma for pollination. The process of this pollen grains reaching stigma is known as pollination. That is the first step. How do they reach? There are two types of pollination. Self-pollination, cross-pollination. In one plant, many flowers are there. Suppose from this flower's pollen reaches this flower's pollen. Both are in the same plant. That is self-pollination. There are two hibiscus plants, one this side and one this side. Here, one flower will be having pollen grains. From this flower to this flower stigma, if it goes between this plant and this plant, it is cross-pollination. Cross-pollination between two different plants. Self-pollination, in the same plant, in the same flower, it comes as self-pollination, or else in the same plant, two different flowers, same plant. That is also self-pollination. Now we need some pollination agents. Let us look into those agents. First is thing, insects. Insects come in search of nectar. Where do they come? Attractive flowers. So the flowers which will be having attractive petals, those flowers are pollinated by insects. They come in search of the that look, that color. The insects get attracted, that scent, that nectar. Insects get attracted. So insects will pollinate in such type of plants. So their back gets sticked for this pollen grains. The pollen grains are easily carried away by the insects. And when this insect lands on another flower, the stigma will be very sticky to collect pollen. If an insect comes and lands, the pollen will immediately touch stigma and pollination will occur. Now that we know insects will is one pollinating agent. Wind, for some trees like cones, for that trees wind will pollinate. Water also will pollinate in few trees. Pollination agents we saw: water, wind, and insects. Let us go to fertilization. It is the next step. So here our pollen grains have arrived. Here one pollen sac will be there, from the pollen grains will be there. Now that they have arrived to the stigma, they go into the style and then they go meet the ovary. Their ovules will be there. Once they meet these ovules, a zygote gets formed. These ovules will turn into seeds and this ovary will become the fruit. I'll tell you this once again. Pollen grains have come, pollination is completed. Next, fertilization. From here, that pollen should reach that ovule. It reaches, it goes through the style and meets ovule. Once it meets the ovules, the fertilization has occurred. Now this ovary will become the fruit. All this petal, stamen will dry off. This will also fall off. Finally, we will have only ovary that has become the fruit, and the ovules, they have turned into seeds. You got now how this fertilization occurs? Now fertilization also completed. Now we have a seed. Now let's go to the next cycle, germination. How will a seed come into a baby seedling and form into a plant? Through germination. In last chapter only we saw in class 6th we saw about germination. Now let us see the conditions required for germination. First thing is warmth, which we will get from sunlight. Next thing, oxygen from air, and water. It needs water. See, suppose this is a seed. If it gets water, the seed coat bulges off. It falls out so that the embryo can come. So it needs water. And then in air, oxygen will be there. Oxygen is needed for respiration, and warmth. It also needs warmth for preparation of its food. So these three things are required. If you perform an experiment, keep some sprouts, that sprouted seeds, before they sprout, keep them in fridge. They won't sprout because they're having, they won't keep it in water and then keep it in fridge. As they're having no warmth, they won't sprout. If you fully immerse it in water and keep it near the window, then also they won't sprout because they're not getting air. Don't fully immerse it. Pour a bit water every day upon it and then leave it in sunlight, a place where it can get sunlight, air, and then sprinkle water. In that condition only it will sprout because it has got its warmth, it has got its water and air. Now germination also completed. Now seed should disperse. Now why seed dispersal is a very important topic? Because if seed dispersal doesn't happen, all the seeds will be near the mother plant. If they are near the mother plant, how will they grow into a healthy plant? Only if they get dispersed, they far and wide, then they can grow happily without any struggle for space. So seed dispersal is very important. Let us see our agents of seed dispersal. First thing is animals. When animals go, there will be some seeds which are which have spines. They get stick to the fur of bears, and when they go land somewhere in the soil, those seeds will fall off. So animals are also playing an important role in seed dispersal. Next, birds. Suppose a fruit is there. A bird will eat that fruit and again it spit it somewhere. If it goes and reaches the soil, again that seed will sprout. Birds, animals, and water. Coconut seeds are dispersed through water. Wind. Have you heard about dandelion? Dandelion is a very round flower. It will be having white. It will be easy to. They are very light in weight. That's why wind will help them in seed dispersal. Dandelions. Now in dispersal, we have been completed of water, wind, animals, birds. And the next is ballistic. Ballistic. Now in this type, what happens is that, suppose if you see pea pods in a pea, 1 minute, in a pea, that pods, once it gets dried, it opens up and falls out. That is known as ballistic. It is drying up and falling up. That is also a seed dispersal. And in some fruits, after they mature, they fall off. That is ballistic. Automatically with the plant itself, they are falling after they immediately get matured. That is ballistic dispersal of seeds. We have completed pollination. What is pollination? The transfer of pollen grains to stigma. Fertilization. From the pollen grains, after they reach the stigma, a zygote should form. That is fertilization. In germination, after we have completed fertilization, one new fruit is formed. And then that fruit seeds. Again, they have to turn into a new plant. So, from a seed to a seedling, it is known as germination. Now, seed dispersal is very important. Then comes the seed dispersal agents: wind, water, animals, birds, and ballistic. So, these are the different types in sexual reproduction. Now, we are going to see class 7 and class 10, nutrition. Now, basically, what is this nutrition? Nutrition means obtaining food and utilizing them for the survival and growth and repair. What is nutrition? Nutrition means obtaining food and utilizing them for their survival and growth and repair. What are nutrients? Nutrients are constitute of the food which provide nutrition. We know what are nutrients, we know what is nutrition. Now we are going to see nutrition in plants. There are basically nutrition is of two types: autotrophic nutrition and heterotrophic nutrition. What is it? Autotrophic nutrition and heterotrophic nutrition. Heterotrophic nutrition. Now, autotrophic nutrition. They can prepare their own food. Suppose autotrophs use autotrophic nutrition. They prepare their own food. They don't depend on other organisms for their food. Heterotrophic nutrition. They depend on other organisms for their food. Suppose humans and animals. We depend on plants for our food, directly or indirectly. If indirectly, for a lion depends on deer for its food. What does deer eat? It eats plants. So indirectly we are depending on plants, or directly we are depending on plants. We are depending on other organisms. That is heterotrophic nutrition. Heterotrophs and autotrophs. Now plants. Plants perform which type of nutrition? Plants perform autotrophic nutrition, and plants are autotrophs. They are autotrophs. Now these autotrophs, they prepare their own food using prepare their own food and synthesize their own carbohydrates. Synthesize means produce. Now photosynthesis is one process by which they prepare their own food. Photosynthesis. Photo means light. Synthesis means produce. Using light, they are producing something. Photosynthesis. It is a photochemical reaction. We know photosynthesis is a chemical reaction. What is photo? Photo means light. It's a light which uses light. So photochemical reaction. We got a brief information about photosynthesis, and plants are using this photosynthesis. How do they prepare? And what are the materials required? How do the materials go? Where does this photosynthesis occur? What are the steps involved? We'll see into it. First, let us go into the materials needed for photosynthesis. Materials needed. Materials needed. Plant needs sunlight. It needs carbon dioxide, water, sunlight, carbon dioxide, and chlorophyll. It will already be present in plants. Chlorophyll. It's a green color pigment. What is chlorophyll? It's a green pigment present in most of the green plants. Now I forgot to tell you an important thing. All plants are not autotrophs. Only green plants are autotrophs. Only green plants use autotrophic mode of nutrition because green plants contain chlorophyll and chlorophyll is important in photosynthesis or to prepare their own food. Now we saw materials needed. Let us see our experiments. What if it is not there? How do we prove that sunlight is required? First thing, after photosynthesis, starch is produced. Starch is produced. When this starch reacts with iodine solution, it turns bluish black. It turns bluish black. This concept is clear. If photosynthesis occurs, starch is produced. When the starch reacts with iodine, it produces bluish black. Suppose we have to prove that sunlight is required. Take a potted plant. It is an experiment. Take a potted plant, keep it in a dark room for some days. Starch content will reduce as it cannot perform photosynthesis. Now use that sunscreen sheet, which means it obstructs sunlight. Use that sunscreen sheet, place it upon the leaf. Now keep the leaf in sunlight for some time. Now the leaf does photosynthesis and produces, but the part where it doesn't get sunlight, photosynthesis won't occur. Suppose this is our leaf. We are keeping a sunscreen sheet on our leaf. That sunscreen sheet, say it is L. This is another sunscreen. Only L gap is left. When we check for starch, everything will turn bluish black. Sorry, everything will turn bluish black except this part. Except this part, everything will turn bluish black as our sunscreen is obstructing it. So sunlight won't happen. If sunlight doesn't happen, starch won't be there. If starch is not there, it doesn't react with iodine. No bluish black. So if a photosynthesis has occurred, the leaf will turn bluish black when we pour iodine. That is one another experiment. We'll discuss about that experiment later. Starch experiment. Main thing is when we perform that starch experiment, that sunscreen sheet obstructed area won't turn bluish black. Now carbon dioxide. Now this is one more experiment. This is Mohl's half-leaf experiment. So what is this experiment? A leaf is there from a potted plant. Keep it like that only, but cover it using one flask. One flask, you're covering it half. Half of the leaf is left, half of the leaf is inside this flask. Inside this flask, keep potassium hydroxide. Potassium hydroxide. Now this potassium hydroxide will absorb, what will it absorb? Carbon dioxide. As it is absorbing carbon dioxide, there will be no carbon dioxide. What happens if carbon dioxide is not there? No photosynthesis occurs. When photosynthesis doesn't occur, starch production? No. Iodine when it reacts, it doesn't turn bluish black. So if you check this portion of the leaf, it will not turn bluish black. Hence we can prove that carbon dioxide is essential, sunlight is essential, water is also essential, chlorophyll is also essential. Now for this and all, wait, we don't have experiments for water, but we know that it is essential. Now let us go into what are the byproducts of photosynthesis. The byproducts of photosynthesis are, byproducts means end products, oxygen and starch. starch in it's glucose. Now glucose, when it is stored, it is known as starch. Stored form is known as starch. So when photosynthesis occurs, the starch is formed and oxygen gas is released. When starch is there, if we pour iodine, it turns bluish black. This is clear. Now we have to prove how is oxygen evolved. Now how oxygen is evolved, we are proving using hydrilla experiment. Now what is this experiment? This a container. In the container we have filled some water. Now a funnel. We are inverting the funnel. Inside this we have hydrilla plant. 1 minute, I'm drawing that hydrilla plant. Or any other water plant also you can take. Water is filled in the funnel also. Actually the water level should be more than the funnel stop. So water level will be this much. Now a test tube. A test tube full of water, we are inverting it. If you keep this setup in sunlight, it will hydrilla plant will perform photosynthesis and oxygen bubbles will be evolved. So oxygen is evolved during photosynthesis. That can be proved using this experiment. Now oxygen experiment we've seen carbon dioxide experiment and sunlight experiment. And one more thing was John Priestley. He is the person who discovered oxygen. Yes, he discovered oxygen. So this John Priestley, what did he do? He performed a series of experiments to find out there's some gaseous. Actually, John Priestley discovered, another scientist named it. What experiments did he perform? That oxygen is present. When he was studying, this happened when he was studying, there was no light. So he on one candle. Due to heavy wind, due to force of wind, the candle blew off. So he wanted his candle to blow. What did he do? He kept it in a jar, that candle. He thought that no obstruction of wind. So my candle will blow as he kept it in a jar. But what will happen? As no oxygen, the candle will not blow. At that time, he did not know oxygen. He was just like, okay, one gas will be there, he discovered, and then one more person named it. We know that. Let's come to our experiment. What he did, he kept that candle inside the jar, but the candle did not burn. He was shocked. I have obstructed air, but why is the candle not burning? There were some rats in his house roaming. He took one rat, kept it, and placed the candle. Now he kept. Now what will happen? Without oxygen, rat also won't survive. The rat also is dead, and the candle also did not burn. What happened? Now next he performed one more series. He had another rat. He kept one more rat. We removed the previous rat, and what he did was he kept one potted plant. He kept one potted plant inside that setup. Now this time what happens? In the potted plant, what does it give out? It performs photosynthesis and gives us oxygen. This is our setup. Wait 1 minute. One rat is here. I don't know. One rat is here, and then one candle. One candle is burning. That when this was there, the candle blew. Because oxygen is released, candle blew. And rat also stayed alive for some more time. As plant cannot produce that much oxygen for a rat to survive and the candle to keep on burning. It produces a bit oxygen so that this rat survived for some more time. Now what he concluded that something which is needed by these two things, something which is needed for candle to burn, something which is needed for a rat to stay alive, is given out by plants. This he framed using his series. After some more years, other scientists came and named as this one is oxygen. Now we got to know about this Joseph Priestley's series of experiments to find oxygen. Now Joseph Priestley experiments we saw we saw that Mohan's half-leaf experiment, that sunscreen experiment. Now what we should do? How do they reach? Suppose we need carbon dioxide, we need water, sunlight. Now how all they are reached to the place where photosynthesis occurs? In a plant, roots will absorb. If you see, roots will absorb that minerals. That is through stem, the water is taken and the water reaches through stem. As it is taken, it reaches the place of photosynthesis. We know through conduction we are going to reach water is we have got water. And stomatal pores. In sixth class we have seen stomatal pores. So those stomatal pores facilitate gaseous exchange. During photosynthesis also, carbon dioxide is taken through those stomatal pores. How is carbon dioxide taken? From stomata. How is water taken? From the root hairs. Water over carbon dioxide over and sunlight. Chlorophyll is a pigment, a green color pigment, which traps sunlight. So sunlight is taken from this green color pigment. Now we know what and all is taken, how is it taken. Now let us see where does this occur, and let us see the chemical equation of photosynthesis. Now what should we see? We have to see where does this occur. In leaves, where stomata will be there. Stomata will be present, in leaf, chloroplasts will be there. Chloroplasts. Now these chloroplasts contain chlorophyll. They contain chlorophyll. So in this chloroplast, a whole photosynthesis occurs. Now they are present in the leaf. Where are they present? They are present in the parenchyma of the leaves, and also near stomata cells. Also these are present in parenchyma of the leaf and near stomatal cells. Now chloroplast, what is it? It is a three-layered, membrane organelle. Membrane organelle. It has three membranes. Two membranes are like that. The third membrane is folded into disc-shaped structures known as thylakoids. Now, so the third membrane is folded into disc-shaped structures known as thylakoids, and a set of thylakoids is known as granum. This is present in the chloroplast only. Chloroplast is basically like this. Like in this chloroplast, it is present, thylakoid. Now in this thylakoid, we have a chlorophyll. Now what does this chlorophyll do? It absorbs sunlight. Now sunlight will be, let us list down the steps. Chlorophyll, sunlight is absorbed. Sunlight is absorbed through chlorophyll, and chlorophyll has got that solar energy. It has got that energy. I'm sorry, no space. Then now that chlorophyll has become active in that place. Now what will that energy be used? It will be used for splitting water molecules. Now the carbon dioxide which is entered will combine with the splitted water molecules, carbon dioxide, and then it forms carbohydrates. And what is the end product? The end product will be oxygen. Of course, carbohydrates are also end products. I'll revise this. First thing is that chlorophyll absorbs sunlight. It has got that energy. With that energy, the water molecules get splitted. And to that splitted water molecule, carbon dioxide reacts. It forms carbohydrates. Carbohydrates mean that glucose it forms. And the excess oxygen is released out. So, if we form a chemical equation, a balanced chemical equation, it will be like this. Carbon dioxide plus water gives you, in the presence of sunlight and chlorophyll, gives glucose C6H12O6 glucose plus O2 in the sense oxygen. So carbon dioxide plus water in the presence of sunlight plus chlorophyll gives us glucose plus oxygen. This is the balance equation for photosynthesis. First thing we covered is materials types we saw, autotrophic, heterotrophic. Materials required also covered. Then we saw all those experiments needed. What are the experiments we did? One more experiment we forgot is the starch experiment that I'll show you now. The materials required we saw, the experiments we saw, and then how do they reach? That also we saw. Occurrence. Where does it occur? It occurs in chloroplasts. Where are chloroplasts present? In the parasite parenchyma and near guard cells. In chloroplast, it is a three-layered membrane. One membrane is folded into disc-like structures like thylakoids. A group of thylakoids are known as grana. Now that we saw the steps also. So what are the steps? First thing is sunlight is absorbed by chlorophyll. Solar energy is used in splitting water molecules. That splitted water molecules is combined with carbon dioxide, which forms carbohydrates. And then excess oxygen is released. This is the balanced chemical equation for that. 1 minute. This is the balanced chemical equation for that. And now that we have seen all steps, occurrence, how do they reach? The last thing is factors, importance, and then our starch experiment. What are those things left? First, we'll go with a starch experiment. First thing is basic starch experiment we're doing for every leaf. In the sense, in every experiment, starch experiment will be included. What do we need? First, we should take a leaf. This leaf, we should boil it in water for some time. Few things which are soluble in water gets dissolved and they get breaked off as we are boiling it in water. Boiling it in water. And next, what we should do? We should take this same leaf, and keep it in alcohol solution. This is a methyl alcohol solution. I'm sorry, this is a test tube. In this we have our alcohol and also our leaf. This we should again keep it in water and burn. Do you know the reason why we are not burning alcohol directly? It is highly inflammable. That's why we are keeping it in water and then burning. Now what happens? Most of the cell organelles gets break down and it loses its color. The main thing why we are performing this experiment, why we are boiling it, so that it loses its color, and then if you apply starch, you can see the color has changed. Now we have done this, color is lost. Now what we have to do? We have to wash it. After washing, keep it in a petri dish. Petri dish is a dish like this. Once you keep it, and now you shall pour iodine solution, 1 or 2 drops iodine solution. Now if starch is there, the leaf turns bluish black. This is a starch experiment. We have used the starch experiment in that carbon dioxide experiment and sunscreen experiment. This is all about starch experiment. Now let us see the photosynthesis importance. And also factors affecting photosynthesis. So materials needed, what we saw: water, carbon dioxide, chlorophyll, temperature. Sorry, these are not materials required, it's factors. These are the materials: water, carbon dioxide, chlorophyll, temperature. And I think these many are affecting the process of photosynthesis. Now water, even if the rate of water, actually plant uses a very less amount for photosynthesis, but even if there's some change in the amount of water they get, photosynthesis can't occur. And so at certain temperature only photosynthesis will occur. At all temperatures it won't occur. So temperature also affects photosynthesis. Water also. And carbon dioxide concentration. It should be there's one thing. If it is in this much quantity, plant will take. If it is in excess or if it is low, there will be a changing rate in photosynthesis. Light also is important. Light that we get from sunlight, it also affects the rate of light. And chlorophyll is very important. If chlorophyll is not there, photosynthesis won't occur. So that is also a factor affecting photosynthesis. So these are the factors affecting photosynthesis. Now if you see the importance of photosynthesis, photosynthesis is the process by which plants prepare their own food. So it helps plants in preparing their own food, and by this process only now every living organism gets its food. How? See, if plants prepare their own food, other animals will eat plants. If plants survive, other animals can also eat plants and they will also survive. So it is the main essential process which keeps life on earth. So photosynthesis get that much importance. And one more thing, these plants which do photosynthesis, after some time they get died, they get buried. That energy trapped in other plants. See, now plants have trapped some energy, they've performed photosynthesis. They get died, they get buried. They, after certain heat and pressure, they convert into fossil fuels. This is the main thing of photosynthesis, is that it is the main source which keeps life existing on earth. And then for that fossil fuels also. In fossil fuels, the photosynthesis in plants which has occurred, energy is formed, and then when they get buried, this energy gets converted into fossil fuels. Now one more thing I forgot is that why leaves are only the site of photosynthesis. See, leaves have a very big lamina, a big surface area to trap sunlight. So they are the site of photosynthesis. And one more thing is that they have many chloroplasts. That is also one reason. And easily when we prepare a leaf through the vein-like network, it can be easily sent to other parts. From those veins only, from those veins of a leaf, it is sent to a stem. So veins are also there, and it has a very big lamina. Afterwards, what else it is? It has chloroplasts. So these are some features. It also has stomata for gaseous exchange. So leaf is the site of photosynthesis. Now that we have seen the types, autotrophic, heterotrophic, materials required, experiments, and how do these materials reach the plant, occurrence, where does it occur, steps required, the steps involved, importance of photosynthesis, that starch experiment, factors affecting. Now we are going to see class 7 and 10, respiration. Respiration in plants. What is respiration? Respiration is the process of breakdown of food to release energy. I will write down the chemical equation. From that, we can get to know the raw materials. So, food plus oxygen. Food is glucose, C6H12O6 plus oxygen. So, oxidization of food takes place, which gives us carbon dioxide plus water plus ATP. ATP are the energy currency, they transfer energy. So they are also known as energy currencies. The full form of ATP is adenosine triphosphate. Adenosine triphosphate. This is the full form of ATP. So when oxidization of food takes place, carbon dioxide, water and ATP are released. So the raw materials are glucose, which is the food, plus oxygen. Where does this step occur? This step occurs in mitochondria. Mitochondria. Where does this step occur of respiration? It occurs in mitochondria. Suppose this is a cell. This is a nucleus. Here we can find a mitochondria. Here our respiration takes place. Now let us see the respiratory organs. In plants there are 3 different respiratory organs. First thing is stomata, next is root hair, and the third is lenticels. We'll go with stomata first. So this opening, it is known as stomatal opening. This gap you can see, this is the stomatal opening. And these 2 bean-shaped cells are known as guard cells. They open and close. This is the epidermal cells. So this stomata is found in the lower side of the leaf. It facilitates gaseous exchange for both respiration and photosynthesis. For respiration, oxygen goes in and carbon dioxide comes out. Next is root hair. This is a root, it's below soil. So these are the soil particles. Between there will be some air spaces. In those air spaces, we can find oxygen. So this tiny root hairs grab that air from there. So this is through root hairs how respiration takes place. Now in herbs, we can find green and tendy stem. For that we have stomata. In shrubs, there is woody stem. For that bark, there are tiny pores they look like dots. They are known as lenticels. What are they known as? They are known as lenticels. So these, they're tiny pores. They help in gaseous exchange. Now let us find about this nematophores. Nematophores. What are these? These are for mangroves. The mangrove roots are known as nematophores. Mangroves grow in salty water, wetlands. They are inside the water now, so they need oxygen. So these roots grow out upwards. From down into the soil, they grow upwards. They are known as nematophores in search of oxygen. Also, breathing roots also we can tell them. Sundarban delta is the largest delta of nematophores. Now we have completed respiratory organs, which is stomata, root hair, and lenticels. Then raw materials, glucose and oxygen. And then we have to see the steps. First thing, oxygen enters and it combines with this glucose. After that, oxidation of food takes place and it breaks down into carbon dioxide, water, and ATP. These are the 3 steps you can take. And now let us find the difference between respiration and photosynthesis. Just a minute, I'll erase these drawings. Then we can see the difference between respiration and photosynthesis. The major difference are the products. With the raw products of photosynthesis are the end products of respiration. So this is our photosynthesis process. Photosynthesis. And here is a respiration. Respiration. What are the raw materials for respiration? It is food, which is glucose, and we have oxygen. What are the end products of respiration? We have carbon dioxide, water, energy. So if you see in photosynthesis, this energy is taken. Solar energy is taken. Carbon dioxide and water is also taken. These are the raw products of photosynthesis. And what comes out is food and oxygen. Whereas for respiration, it's reverse. This is the raw materials. This is the end products. This is one major difference we can find. And one more thing is, respiration occurs all time. Photosynthesis occurs only during daytime when light is there. Photosynthesis mainly occurs only in plants. Respiration occurs in all organisms. So this is the difference between photosynthesis and respiration. We saw the steps, raw materials, and respiratory organs. Now we are going to see plant tissues of class 9. First, what is a tissue? A group of cells with similar size, shape, origin, and function. A group of cells with similar shape, size, origin, and function are known as tissue. Now we have different types of tissues in a plant. First, we have meristematic and permanent. What do we have? Meristematic and permanent. Permanent tissues. 1 minute. Now, this is the dividation in plant tissues. In meristematic, we are going to have 3 different types, which are intercalary, lateral, apical. 1 minute. Intercalary, lateral, apical. These are the 3 types in meristematic tissue. How is meristematic tissue basically differentiated? Based on their position. Based on their position in a plant, the meristematic tissue are divided. Let us draw a rough diagram of plant, and then we will check into this meristematic tissue. Just a minute. Suppose this is a plant diagram. This is a stem. This will be a roots. Here, if it is present at the tip of the stem, means at the top or at the bottom of the root, here it will be present on the tips of the stem, roots, and branches, that is apical meristem. Dividation of apical meristem. I forgot to tell you the two main important things. Why is it divided into meristematic and permanent? These meristematic tissues are capable of dividing. Meristematic cells are capable of dividing, whereas permanent tissues are not capable of dividing. They're capable. They have thin walled. They're thin walled, compactly arranged, very closely arranged. So these are the major features of meristematic. Now we are going to see its positions. Tip of the stem, branches, roots. It is apical meristem. Next comes, if apical meristem divides, it increases in the length of the plant. It is the primary growth. Next we have lateral meristem. Lateral meristem, if you take, this is the stem, okay, it is present along the sides of stem, root, etc. This is the lateral meristem. If this increases, then the width will increase. It is the secondary growth. Intercalary. Suppose I am taking one node, leaf base, nodes, internodes. Here and now we can find intercalary. Nodes, internodes, there are no between. Now intercalary, it also sounds something, it's inter. So that intercalary we can see in these places. Apical, intercalary, lateral. Wait, let me write the spelling of intercalary. Intercalary. Apical, intercalary and lateral. These are the three different things in meristematic. First main feature is that they are capable of dividing. Second thing, they are thin walled, compactly arranged, and they lack intercellular spaces. Now what is this intercellular spaces? Between two cells, sometimes spaces will be there. Sometimes they're so compactly arranged that there are no spaces. So no intercellular spaces. Whereas you can find intercellular spaces, or one more cell is there's some intercellular spaces. In some cells, some tissues, we have intercellular spaces, whereas in meristematic we are not going to find any intercellular spaces. This is about meristematic, and it's of three types: lateral, intercalary, apical. Based on the position, it is divided. Now apical meristem, it is present on the tips of stems, roots, branches. Lateral meristem, it is present along the sides of the stem and branch. Apical meristem's dividation causes the increase in the primary growth. Lateral, it causes an increase of secondary growth. Intercalary, it provides strength as it divides. Now permanent. Permanent is very vast. Here if you see in permanent, there are two dividations. Which are those? Simple permanent and complex permanent. Simple permanent and complex permanent. In simple permanent, again we have two types. One is protective, and ground tissue. Supportive or ground. Supportive or ground. Support. And protective, we have again three types: epidermis, cork, wax. And supportive, parenchyma, collenchyma, and sclerenchyma. This is C, sclerenchyma. In complex permanent, we have basically two: xylem and phloem. Okay, see, first let us know the features of permanent. They have intercellular spaces. They are differentiated on basis of their composition. And one more thing, when these meristematic tissues lack the ability to divide, and acquire a permanent structure, size, shape, They are known as permanent. When these meristematic tissues lose the ability to divide and acquire a permanent structure, size, shape, they form permanent tissues. Okay. Now, in permanent tissues, we have simple permanent and complex permanent. In simple permanent, we can take a group of pens. Suppose we are going to keep only meow pens in one box. It will come under simple permanent. All these meow pens have some function in common, which is writing, and they are of the same type, same cells. In simple permanent means same cells. In complex permanent, we are going to keep it's a box of pens, but meow pens, exo pens, link pens, different types of pens. In complex permanent, all of different brands. They are different cells, but working together with the same function. Pens means they have the similar function, writing. So that's the major difference. You can think of simple permanent and complex permanent. They are made up of the same type of cells working together with a similar function. They are made up of different types of cells working together with the same function. Now that we have got to know about the differentiation. Differentiation means the process by which meristematic tissues lose the ability to divide and acquire a permanent structure, size, shape. It is known as differentiation. We know differentiation. We know in simple permanent there are we know what is simple and complex permanent. Now in simple permanent, it's protective and supportive. In protective, epidermis. Now this epidermis is the major covering of a plant. It is epidermis. Epidermis has cuticle. Cuticle. Now this cuticle in epidermis, it's like a tissue which covers the stomata. Hence respiration gets reduced. It reduces the loss of water from the leaf. The cuticle in epidermis. It's an outer protective covering. Now we have cork. Cork. If you see in many trees, you won't find epidermis. Instead, dead cells are there. A bark we call it. That is also known as cork. So that is the cork main thing. It also helps in reduce of loss of water. It prevents the entry of pathogen. It facilitates gaseous exchange. It bears lenticels for gaseous exchange. Now wax. Wax is the waxy coating. Now see in leaves. Take a normal plant's leaf. Any plant's leaf growing on the land, and keep it in water for some time. It will decay. Whereas aquatic plants, they're in water for most of their lifetime. Almost lifetime only they're in water. But also they're not decaying. Why? Because they're having this waxy coating. Waxy coating protects them and prevents them from decaying and destroying. Now that we have seen the different types in protective tissue, which is epidermis, cork, and wax, we are going to see into supportive tissue. Supportive tissue, we have parenchyma, collenchyma, sclerenchyma. Now parenchyma. This is present in the major softer parts of a plant, and parenchyma in leaf contains chloroplasts. It contains the parenchyma in leaf. It will be containing chloroplasts. Chloroplasts. So it is known as chlorenchyma. And then some in aquatic plants, parenchyma has air spaces known as aerenchyma. Now we have seen its different names. Let us see into its arrangement. They are thin cells. They are living cells. They are rounded, circular, or polygonal in shape. They're thin cells. They're thin walled. They're rounded, circular, or polygon. And one more thing we can see, they're living cells. We have gotten about parenchyma's composition. Let us see into parenchyma's functions. It stores food. It is the seat of photosynthesis in chlorenchyma. In leaves, it is the seat of photosynthesis, chlorenchyma, as it has chloroplasts. It stores food. Also in aquatic plants, it has air spaces. So these are the different functions of parenchyma. And collenchyma. Collenchyma, the structure, let us see. Collenchyma, they are they're living cells. They're thickened at corners due to the presence of cellulose. They're living, they're thickened at corners due to the presence of cellulose. They have no intercellular spaces. They have a bit less intercellular spaces. Parenchyma has many intercellular spaces, less, and in sclerenchyma, no intercellular spaces. Collenchyma's function: elasticity, tensile strength, rigidity. Rigidity means hardness. Elasticity means that pulling, stretching. Tensile strength means that strength which helps the plant in being elasticity. That is known as tensile strength. These are the different things in collenchyma. Now in sclerenchyma. 1 minute. Now in sclerenchyma, what is its structure basically? Sclerenchyma is present in fibrous plants. It gives that fibrous plant mechanical strength. This sclerenchyma, it is thickened due to the presence of pectin and cellulose. It is a dead cell. Also, it has what do you say? No intercellular spaces. It provides mechanical strength. The sclerenchyma we can see in fibrous plants. These are parenchyma, collenchyma, sclerenchyma. Under simple permanent. Now let us see complex permanent. 1 minute, I shall erase. Simple permanent is over. Complex permanent. Now main thing is xylem and phloem. First, let us learn about xylem and phloem. First, xylem. Both have some features in common, so I'll explain them simultaneously. In xylem, what are the main things? Tracheids, xylem vessels, and then xylem vessels, xylem tracheids, xylem parenchyma, xylem fibers, xylem tracheids, and here we have sieve tubes. We have after sieve tubes, what do we have? I'll write it down. Companion cells, sieve tubes, sieve cells and sieve tubes, and then phloem parenchyma. We have phloem fibers. Now these functions of these are quite similar. See in tracheids, they have tapering ends. Let us see the composition. They are thick cells, they have tapering ends, and they transport water and minerals. What transports water and minerals? Tracheids. Now if you see in a phloem is basically the conduction of the food prepared by the leaves. Here we have sieve tubes. Sieve tubes also transport the food prepared. So sieve tubes and tracheids are somewhat similar. And these xylem vessels, they are placed end to end and they form a cell cavity. The sieve tubes also, they are placed end to end to form a tube-like structure. Sieve tubes have no nucleus. So companion cells nucleus control the function of sieve tubes. Companion cells, company it gives. It shares its nucleus with the sieve tubes. So basically, the sieve cells are placed end to end. They form a tube-like structure. This tube helps in transporting food. In trachea, xylem. This is in xylem. We have xylem tracheids. Tracheids are thick and they have tapering ends. They help in transport of water and minerals. Xylem vessels are xylem cylindrical cells. They are also placed end to end. Now, parenchyma. Xylem parenchyma provides strength. Phloem parenchyma provides strength. This provides strength for xylem. This provides strength for phloem. And fibers. I'm sorry. Fibers provide strength. Xylem fibers provide strength to xylem. And phloem fibers provide strength to phloem. And parenchyma is storing food. As you saw in simple permanent also, we had parenchyma. That also stores food. So here in phloem and xylem also, parenchyma is used to store food. Here in xylem, it stores food in the form of starch and fat in the form of tannin. Now that we have seen everything, the major differences. In xylem, everything is dead except parenchyma. In phloem, everything is living except phloem fibers. One difference. It helps in the transport of water and minerals. It helps in the transport of food prepared by the leaves. The complex permanent also we have completed. The plant tissues, main thing is meristematic and permanent. In meristematic, we saw apical, lateral, intercalary. In permanent, simple permanent, and complex permanent. In simple permanent, we have supportive and protective. In protective, we have epidermis, cork, and wax. In supportive, we have parenchyma, collenchyma, sclerenchyma. And in complex permanent, we have xylem and phloem. In xylem, these are all the things which contribute to xylem, and these are the things which contribute to phloem. And we have seen the differences also. Now we are going to see class 10, transportation. What is transportation? Transport means moving from one place to another. So in plants, the water and minerals of the food moving from one place to another. In human body, we have blood. Blood transports oxygen, blood also transports the digested food. In plants, there is no separate system. These xylem and phloem help in transporting of water and minerals. Now basically how they are absorbed also we'll see here only. First thing, how water and minerals are absorbed in the root. Water though it will get absorbed through osmosis and diffusion. If you see minerals, diffusion and active transport. Now what is the difference between osmosis and diffusion? What is diffusion? Suppose water. There are many water molecules in the soil. This is the soil. This is the root hair. Let's come like this. There are many water molecules in the soil. But whereas in the root there are only few water molecules. From a higher concentration to a lower concentration. The movement of particles from a higher concentration to a lower concentration is known as diffusion. Whereas in osmosis, the movement of particles from a higher concentration to a lower concentration when a semi-permeable membrane, semi-permeable membrane. When a semi-permeable membrane is there and diffusion takes place, we call it as osmosis. For osmosis, semi-permeable membrane is needed. For diffusion, there is no need of this semi-permeable membrane. Directly from higher to lower concentration it will move. And the minerals, they are dissolved in suppose magnesium, phosphorus, etc. These minerals, calcium also, they are dissolved as ions in water. As ions, they are dissolved, and these also are taken into the plant by diffusion and active transport. Active transport is that suppose minerals are less outside the root. There are many minerals in the root, but already it has observed, but there are very few minerals in the soil. Through active transport, it acquires energy. From this energy, from a lower to higher also it is able to pull. This is known as active transport. So, water and minerals, how are they absorbed? Minerals, how are they absorbed, we saw. Now, how are they transported? They are transported using this xylem and phloem. We have learned this in plant tissues of class 9. So, basically, xylem transports water and minerals from the root to other parts of the plant. Phloem, it transports food from the leaves to other parts of the plant. This is all about transportation. First thing is that how do roots absorb, that we saw, and next thing is transportation in which they occur, through which they occur, xylem and phloem. Thank you. If you really like our videos, please do like, share and subscribe.